This video comprises of difference between different type of RNAs such as HnRNA, SnRNA, mRNA, t-RNA, r-RNA that become very easy for us to understand, because in this video I will explain all these things on the basis of their diagrams and their names. It become very easy to differentiate between all of them, and in the end of video I also have Q&A related to these RNAs so that you can identify them. You can identify all the RNAs on the basis of their structure very easily, and on the basis of their name you can also tell the difference between them. So let's get started. Hello friends, welcome back to my channel Biology with Agam Singh. So let's get started with the point number 1, or the type 1, that is HnRNA. What is HnRNA? HnRNA is the heterogeneous nuclear RNA. Why it is known as heterogeneous nuclear RNA? Because it contains heterogeneity in its complex, as it contains introns as well as exons. This blue part is a exon, that is a coding part, while the black part is the intron, that is a non-coding part. So why it is known as heterogeneous? Because it is when we talk about the homogeneous, then it is comprising of only a single thing, but this comprises of introns as well as exons. That's why it is known as heterogeneous RNA. One more feature about this RNA is that it is much more longer than other RNAs, because it is the primary RNA that is formed after the transcription. As we know that after transcription, post-transcription modifications are required to make a mature functional RNA. But it is the primary RNA that is not mature RNA, that is not that is the RNA before the post-transcription modification. So this is the primary RNA which is formed after the transcription. Okay, which contains many impurities, means that the exons and the introns. Exons are the coding part, they contain coding part as well as non-coding part, and they doesn't contain N terminals. Means that if it is a 5 N terminal and the third terminal 5 and 3 terminal, but it doesn't contain any type of capping or tailing here. As in post-transcription modification, we see that there are 5 end cap and 3 end polytail. So there is nothing about that. It is just the raw RNA. Okay. So for the HnRNA, we can say that it is heterogeneous nuclear RNA, which is a primary RNA which consists of exons as well as introns. It's for example, its heads are just like that. 5 and 3 ends are, it is unstable RNA actually, we can say it. As such, in this rope you can see that these ends are splitted, means that if I stretch them, they will destroy. As such, this RNA is also destroyed when endonuclease or some enzyme will bind. It will cut it out and destroy it. It is unstable form of RNA. Okay. Now the SnRNA. What is this? Is one the HnRNA. HnRNA. Okay. This is the first one. Now the SnRNA. What is SnRNA? A small nuclear RNA. As the name suggests, small nuclear RNA. A small nuclear RNA. As the name suggests, SN for this. Small nuclear. It is small. That's why it is named small. Why it is named small? Because firstly, it is of a small size. It is just about 135 nucleotides long. 135, only just 135. If we talk about the HnRNA, it is about 4,000 to about 40,000 or 50,000 base pairs or nucleotide long. Means that 50,000 nucleotide long. It's a very big number and such a size. That's why it is unstable also. But in SnRNA, it is a stable RNA. It contains the coding region only. It contains the coding region only. But what is the function of the SnRNA? SnRNA is that RNA which helps in post-transcription modification. As we had studied in post-transcription modification, the three things includes: first, 5 end capping, second is 3-end poly-A tail, and the third one is splicing. So the SnRNA works in splicing, making the spliceosomal complex which works on the HnRNA. Means that SnRNA and HnRNA are interlinked to their functions. Means that the SnRNA will goes to this side if this is an SnRNA 3 to 5, but it mostly doesn't appear alone. It makes a complex with SNRP. What is SNRP? Small nuclear ribosomal proteins. Means that they make complex with some specialized protein for themselves and combine with them and then stay here. Okay, so they make a spliceosomal complex. This is like a ring. When they make a spliceosomal complex, they help in splicing. Splicing is what? In which the exons will remain here, but the introns from the HnRNA are removed. So they will bind with the intron part as such. They will bind with all the intron parts, and what is their function? Their function is very simple. That is, when they bind to introns, they starting cleaving the introns from the main strand of HnRNA and remove it. Means that they bind it and remove it. Now they remove. After removal, they join the exon part. As such, they all removes the intron parts and exon part will remain here. Okay. And finally what we got? Finally we got a mature mRNA. Finally we got mature mRNA, but two steps are also left here. That is, third end poly-A tail and the fifth end capping is left. Okay, but SnRNA works on the principle of spliceosomal complex, which removes the introns. That's it. That make from the small, you also recall that it makes the HnRNA small. As the introns are removed, so it becomes smaller in size. That's why it is also known as small nuclear RNA. Okay, so both of these terms are clear, I think so. Now come to the third one, that is mRNA. That is the byproduct, or we can say that it is the post transcriptional modified RNA or the mature RNA. It is known as mRNA. Why? Firstly, I will rub this. We will make the mRNA here. Okay, it's an mRNA. Okay, it's contain 3 and 5 end. It is mRNA. Why we is saying at mRNA? Mostly we will say that it is messenger RNA. It is a messenger RNA, but we can also say that it is a mature form of RNA. Mature. Why we are saying it mature? Because it had done with the it had done the post transcription modifications. What is that? Means that a mature mRNA contains what? If I change the terminal here, this is the third end and this is the 5 end. Okay, it contains 5 end cap. 5 end cap. Firstly, make an example. That is, when I take this rope and this way, when I stretch it, it becomes reattached and become unstable. But when I make knots here, then when I stretch it, it doesn't become unstable and doesn't degrade. As such, when I also made the node in the second terminal, as it is also this like unstable, so when I make nodes here, it becomes stable. So this mRNA contains these type of nodes in the form of 5 end cap and the third end poly-A tail. Poly-A tail, A is for what? Adenylic acid. It contains about 200 to 250 adenylated chain continuously. Means that one A is connected and then so on, it is connected on up to 200 to 250. Okay, this make what? that make, as I saw the example in the rope, this make stability. The poly A tail make it stable. It is as when the poly A tail is not present, then endonuclease will bind with it. And when endonuclease will bind, what is the function of endonuclease? Endonuclease cleaves the RNA or the genetic material. Means that endonuclease is an enzyme which cleaves the RNA or DNA in small fragments and destroy it. So due to the poly A tail, it becomes stable, so that endonuclease will not bind here and it doesn't degrade. Okay, in the 5' tail, it also makes the endonuclease to doesn't bind. And one more function of 5 and poly A tail is that in eukaryotes, if we talk about the eukaryotes, 7 mg, it is made up of 7 methyl guanosine, 7 mg cap. We can also say it mg cap. Why we are saying it? 7 methyl guanosine tail is present, that is recognized by the ribosome, as we had studied in the translation, that in eukaryote 7 methyl guanosine cap will recognize the ribosome binding site and then bind with it to make a ribosome mRNA complex. So this is also helping that. So this is a mature RNA which is stable. Okay, HnRNA is not stable, as it can as it contain many type of exons and introns, and the terminals are also not confined. Here the terminals are chemically modified. So this is a stable RNA or a mature RNA. So relate all them so that you can learn it properly. From HnRNA to SnRNA. HnRNA is a long RNA. SnRNA make it small, as it is small, and it works to make it small. Okay, now mRNA, which is a byproduct, or we can say post transcriptional product formed after the post transcription modifications of HnRNA, means that it totally refined and then mRNA is formed. Okay, these are the three RNA that you have to connect with all of them so that you can recall it very easily. Now the third one, that is tRNA. What is tRNA? As the name suggests, tRNA. tRNA. What is tRNA? T stands for two things. Firstly, understand T for, as we can write the alphabet T. It is just like that. It contains these two sides as such, and this is the another side. If we make it short, then it is the another side. It is just like the small letter T. That's why it is named as T. Another is it is what is the full form of T? T is for transfer. So from the transfer, what is transfer? It transfers the amino acids. That's why it is known as transfer RNA. It transfers the amino acid where? When the translation is done, then these tRNA take out, as such in this example, it take phenyl NH2, phenylalanine. It takes phenylalanine to the ribosomal binding site where it contains anticodons, which will bind with the codons, which are present on the mRNA strand. This is the mRNA strand, and they will bind to the coding strand with the hydrogen bonding. Okay, so what is the specifications of tRNA? Firstly, that tRNA, first T is stand for its shape. It is T in shape, T shape. That's why first thing is that. Second is transfer. It transfers the amino acid so that amino acid will link together with the peptide bond and a protein will form in the translation process. Okay, so this is the tRNA. It contains anticodons in the anticodon loop. It contains 4 loops. It is not interested. It is not important here, but I can tell it. That is DHU loop. It is a DHU loop. This I am telling to this. Okay, it is T-Psi-C loop. Okay, this is anticodon loop, as it contains anticodons, that's why. And it also contains a arm. I had forgot to make it. It contains a side arm here also. So these are the basic structure of tRNA. The fifth one, that is rRNA. Let's make it. Now the fifth one, that is r-RNA. Why it is known as r-RNA? Just make it r-RNA. Why it is known as r-RNA? Because the R stands for ribosomes. It stands for ribosomes. As it is present in ribosome, that's why we are telling it in r-RNA, ribosomal RNA. That's why ribosomal RNA. That's why it is present in ribosomes. So what is the role of ribosome? As we know that we had studies in many places that rough endoplasmic reticulum contains lot of ribosomes on its surface. That's why it is known as protein factory. So the ribosomes play a major role in the protein synthesis. It is a major place where the protein synthesis occurs. So this is the RNA which is useful for the protein synthesis and present in the ribosomes. That's it. Now I have to make a connection between tRNA, mRNA and rRNA. So what is the connection? As I had made the connection between HnRNA, SnRNA and mRNA, that SnRNA will work on HnRNA to make it shorter, smaller, and for the splicing process and make the mRNA. As I said, as I connect the mRNA, tRNA and rRNA in a process known as translation. Ribosome, as we know that ribosome contains rRNA. It contains two subunits, the 30S subunit and the 50S subunit. If I talk about the 70S ribosomes, okay. So it will bind with the mRNA, okay, and mRNA, after that binding of mRNA and making the ribosomal mRNA complex, the tRNA will come and bind to the mRNA codon in a specific place, that is P site. The tRNA will bind with it, okay, with its anticodon loop, okay. So this is the relation in the translation. These are related in the protein synthesis to each other, means that mRNA, tRNA, rRNA both are included in the protein synthesis. So we can make connections from here. Now this is the time to summarize that all type of RNAs with some images, some diagrams, so that you can memorize it for a long time. Okay, starting with the first, that is it contains exons and introns and it is larger in size. What is the RNA? The name of RNA is, guess it out, it is HnRNA. As I told you, it is heterogeneous. Introns and exons are always present in it, and it is so longer. Okay. Now the second one, it is a shorter RNA and makes a complex known as spliceosomal complex with some specific proteins. So what is the name of RNA? Obviously, it is SnRNA. It is now the third one, that is it contains a 5' cap and the third and poly A tail and contain only the coding parts. What is the name of RNA? The name of RNA is obviously m-RNA, the mature or the messenger RNA. Okay. Now the fourth one, it contains a shape of a T, alphabet T like shape, and it contains anticodons in its anticodon loop. What is the name of RNA? It is the t-RNA. It is t-RNA, the transfer RNA or the T-shape RNA. Okay. Now the fifth one, it is present in the ribosomes. It is only present in the ribosome mostly in the cases. So what is the name of RNA and process and works in the formation of protein synthesis? So what is the name of RNA? It is obviously r-RNA. Okay. So these are all the points that you have to list it out. After making the figures of such like that, you just have also to make these type of images, these type of diagrams and answer these, so that you can differentiate between all of them. I hope this video is helpful for you. If you like this video, then give it a thumbs up, and if you have any issue, then you can ask in the comment box down below. And if you haven't subscribed this channel, then subscribe the channel to get more interesting videos such like that. Thank you so much for watching. See you soon.